Melissa, thank you very much, and thanks to the organisers for inviting me to talk. So I'm going to give you a bit of a whirlwind tour in terms of some of the work that we've been doing in schizophrenia, both clinical, and then we'll move to preclinical models that my colleague Jess will present after me. So I put this picture here because when people develop an acute psychosis, it's very much like a storm. It's very distressing for the individual, anxiety-provoking for families, and very difficult to manage. And I'm going to talk about some of the clinical work first, and then about how we moved to some of the preclinical work. First, an acknowledgement of country and acknowledgement of the traditional owners of the land on which we are presenting today. So in this talk, I'll present data on brain changes in psychosis and schizophrenia, which is much of the work that I've been doing over the last 30 plus years. Mapping trajectories is certainly an important way forward. So not just looking at one point in time, but looking at things over time. And that's important not only in terms of looking at brain change, but also looking at clinical changes in our patients, because it speaks to helping us to understand risk factors, resilience factors, and the way that the illness evolves from pre-psychosis right through to the more chronic stages of the disorder. And then really mapping is fine, and it's great to be able to map the brain changes, and we've done a lot of work in that space. But really trying to understand what might underlie those changes is what the focus of my current work is. Trying to understand why there are brain changes, what are the mechanisms at the cellular level. So we'll be diving down into the brain to try and see if we can get a handle at least on some of those issues, and I'll be focusing on things like the importance of metals, particularly iron, in the brain of the patients that we're seeing. And from there, from all that clinical work, we've moved now to preclinical studies to try and get a handle first on those neurobiological changes, but then to see if we can intervene during the developmental maturational process to make a difference to patients. And it may be that we need to be intervening at a much earlier stage than we thought. I talk about bedside to bench research because often we talk the other way around. We talk about bench to bedside, but actually the work that I've undertaken and the questions that have arisen that I've been keen to answer evolved from clinical observation. So going from the left through gradually to the right. And through clinical observation, we started to think about, well, what's happening in the brain of our patients? And I was intrigued to hear from Professor Burdigan this morning about some of the individuals suffering with mental illness that he encountered, because prior to 92, I was in London and working, did my first research study looking at the epidemiology of schizophrenia in inner London. And as part of that, I saw patients or people suffering with schizophrenia in homeless hostels, people living in the streets, people in prisons. And it was a very instructive study because it helped me to get an understanding of the breadth of the presentation of this disorder in all its forms, and really led me to start thinking about the brain in more detail, that we really need to start thinking about what's going on. And our first studies were on cognition. We undertook the first studies using the CANTAB tasks that you heard in Melissa's talks in our patients, and that led to us thinking about which brain areas are involved and implicated. And more recently, we've started to undertake neuropathological studies, looking at studies post-mortem to look at things that are happening at the cellular level. And working with colleagues at the Monash Institute of Pharmaceutical Sciences, we're now looking at receptors at the cell surface and drugs that might modulate those with a view to identifying novel treatments, because we need better treatments than what we have to date. We have treatments, which is fantastic. Certainly, we're in a better space than, say, Alzheimer's dementia, where there we understand the pathology, but we don't have treatments. Here, at least we have treatments, even though we are struggling to understand fully the pathology in the disorder. So what do we know about schizophrenia? Well, it affects young people. We know that. People in the prime of their life, at a stage in their trajectory when they should be at their most productive, when things are just taking off. So very distressing to see young people affected by this disorder at such an early age. Women seem to get it a little bit later. And then again, there's another increased incidence at the menopause. Again, speaking to the notion that estrogen may be protective in terms of the disorder. It occurs across the socioeconomic spectrum. So it doesn't, although environmental factors, as we heard earlier today, are certainly important and can be relevant in increasing the risk. There's a high genetic component, but it's complex. It's a disorder that involves many genes and across the whole genome. So it's often spoken about as a polygenic disorder. And these genes interact with environmental factors to lead to what we see in the disorder. So when we look at genetics, it's important not just to look at genetics, but also to look at the environmental influences on genes, and particularly neurodevelopmentally, in other words, over the course of development and maturation. There is some variation in the incidence of schizophrenia across the globe, and it was thought that there may be a equatorial to polar shift in the incidence, with higher incidence as you move away from the equator, which may speak to the importance of inflammation, infections, etc. But overall, it's fairly modest. And then childhood schizophrenia is rare. If we looked at all the cases in Australia across 1 year, we'd probably get around 50 cases. So this suggests that the disorder is a disorder of adolescence and early adulthood. And that immediately raises the question, well, what is it about this maturational period of time, this epoch, that is important? I was fortunate to be undertaking a sabbatical in Copenhagen, working with the schizophrenia researchers there. And we undertook a study looking at using their case registers, their twin register, and we undertook a study in Danish twins. We looked at a number of risk factors, and we'd heard a lot about risk factors today. So here are quite a few of the ones that you heard about earlier. And we were interested to see which were most relevant in terms of the risk for schizophrenia. Of those, 3 seem to be most important. Genetic risk, so that's the polygenic risk score. But then look at the odds ratio for the other 2, childhood trauma and regular cannabis use. So there was a 4.5-fold increase in the risk in those who had suffered childhood trauma, and an 8-fold increase in those with regular cannabis use. So cannabis is certainly important in terms of what we're seeing in the disorder. And there was a more recent study, a case register study again from Denmark, involving almost 7 million, I think, people. And it looked at the relevance of cannabis in terms of schizophrenia. And again, cannabis is important in increasing the risk for the disorder, and especially in males. So I know there's a lot of interest in the use of cannabis and using it in various kinds of conditions. When we're talking about psychosis and schizophrenia, I'd have to say it's a no-go zone. And we've looked at the brain and what happens in the brain in patients as well as heavy cannabis users, but we can come to that if you're interested. So I mentioned the adolescent period, the early adult period is important. And what's interesting is that not only do you see psychosis in mental disorders, you also see it in neurological disorders. And you see it in neurological disorders that start early in late adolescence or early adulthood. Here, Dennis Velakoulis, a close colleague, looked at all of the published literature on frontotemporal dementia, which is a young person's dementia, usually occurring before the age of 65 in contrast to Alzheimer's dementia. But there were also a number of cases that began in their early 20s. And when you looked at the presentation of those, so the line, that's the ages on the x-axis there. I think I've got a pointer somewhere. The proportion of younger people developing presenting with frontotemporal dementia in their early 20s were significantly more likely to manifest psychotic symptoms. So again, it's something about the brain at this early stage, a still maturing brain, that is important in terms of psychosis and schizophrenia. So it's important, therefore, to have a context and understand normal neurodevelopment, normal brain maturation. And this was a really fabulous paper by Nitin Gogte and Judy Rappaport from NIMH in Washington, where they did serial scans from age 4 to age 21 in healthy children, and they looked at the brain changes. And what you see there, I think, here we go, is this gradual thinning. So as the brain becomes blue, you see the cortex is thinning slightly. And it starts at the back of the brain, moves forward and downward, and finishes in the region of the frontal lobes, so the frontal part of the brain and the temporal lobes. And they're the last to mature in late adolescence, early adulthood. These are the regions that are particularly impacted in schizophrenia, which is really quite interesting. And when you look at what's happening at the cellular level, these are dopaminergic neurons. You can see at birth, they're not particularly connected. By age 6 or so, there's a massive proliferation of connectivity. The neurons are all wiring up. And you see that in the behavior of children. You know, the kids in early childhood are very active and they're using up a lot of energy. And a lot of that energy is directed to the brain because the brain is gradually being molded in quite dramatic ways. And as they move into adolescence and early adulthood, there's a gradual refinement of that dendritic arborization. That tree is pruned. And that pruning process, we believe, is relevant to the neurobiology of schizophrenia, and in fact may be occurring too rapidly in the disorder, as I'll go on hopefully to show. So pruning finishes late in the frontal lobes and other association cortices like superior temporal lobe. That is likely to be important. And this hypothesis, this pruning hypothesis of schizophrenia, was put forward by Feinberg in 1982. And it's only now we're beginning to try and get a bit of a handle on it. So as I mentioned, the frontal and temporal lobes are consistently found to be impacted in schizophrenia, with thinning in those regions in particular. This is a large consortium study, the Enigma study, and it shows that we participated in, we contributed data from the Australian Schizophrenia Research Bank that was headed by Vaughan Carr. And that's been a fabulous collaboration with international researchers. And here you see that the temporal and frontal lobe regions are showing this loss of the brain's grey matter. And here we undertook a study looking at the brain's white matter. Now, these are the connections that connect the grey matter from one region of the brain to the other, the axons from the neurons. And what you see here, and I'll get you just to focus on the left-hand little movie there, we've got an index of the integrity of that white matter. It's called fractional anisotropy. It's the way that the flow of fluid is propagated through those axons. And what you see in the red there is a reduction in that connectivity. There's a reduction in those axonal fibers. The other thing that's interesting about this figure or this movie is that it's across the whole brain. So whilst, yes, we're seeing those changes in grey matter in frontal and temporal lobe regions, when we look at white matter, it seems to be a whole brain affair. And then again, looking at things by age, because it's really interesting to look at how things are changing over maturation and over the development at different stages of the disorder. Here, Vanessa and as in our team looked at the grey matter by age and the changes in the grey matter in normal controls versus patients with schizophrenia. And it looked as though, well, it demonstrates that in the early 20s to 30s, that grey matter is being lost at a greater rate compared to healthy controls. When we look at white matter, that seems to be occurring at a later age. So the grey matter seems to be being lost earlier than the white matter. And we think that may be due to loss of those synapses at that early age in terms of the loss of the grey matter thickness. And then Cassie, who'll be speaking soon, undertook this study looking at looking at an index of connectivity across the brain. She looked at the way that the thinning of the brain was correlated across the brain, if you like. The cortical thinning was greatest in those with treatment-resistant schizophrenia, so the more severe phenotype of the disorder, so those with the more severe illness. But what was interesting was that it was those regions that were connected that tended to show this thinning. In other words, it wasn't random. It was areas that linked together that also showed this thinning across stages and ages. So then we started to think about longitudinal changes, and indeed, what really got us thinking about how the brain is changing dynamically was a study we undertook early on back in the early 90s, which was really driven by Professor Dan Weinberger, and I was fortunate to be working in his lab in the early 90s. And when anyone new, such as myself, appeared in the lab, he'd show us a couple of scans. And he'd just finished a study in schizophrenia that was published in a very prestigious journal, the New England Journal of Medicine. And he'd show us the scan, and there were two things that discriminated who had schizophrenia and who didn't. So I guess I've already given you the answer, but the twin with the larger ventricles, the fluid-filled spaces in the brain, were the ones that had or showed schizophrenia of the twin and co-twin. The other thing that was prominent was a loss of volume of the hippocampus. The hippocampus is important to memory, to behavior, and to connectivity with the frontal lobes, and has been implicated in schizophrenia. And we thought, well, given those findings, and given that this study suggests that these two indices may be biomarkers, if you like, of the disorder, we should have a look at that. And I was fortunate to be to come back to Australia in the early 90s when Pat McGorry, that you can see there, and Alison Jung, who's in the audience, of course, and this is an early picture in the early 90s when you were just getting started with the high risk for psychosis research. And I thought, well, this is a great opportunity. If we get brain scans at that time in people who are at high risk and then follow them up, surely we'll be able to use the brain imaging to identify who is going to develop the disorder. At least that's what we thought, right? So these were the criteria for those at high risk. Alison, you're going to tell me that there are more recent studies than the ones I'm quoting here as well, but you can tell us about that later. At the time when we were doing the study, almost 40% were making the transition to psychosis. So 40% of those identified by Alison and Pat who were deemed to be at high risk for psychosis were converting to psychosis within about 18 months. So we were doing brain scans before anyone became ill, and then after when they did, or we followed up those that didn't as well. And here again, Dennis Velakoulis started to look at the hippocampus. We thought, well, if this is a marker, then surely we should be able to measure the hippocampus in those that are at high risk, and the hippocampus should be able to point to those who are more likely to develop schizophrenia or psychosis. So there were 500, almost 500 people in this study, so 1,000 hippocampi almost. Dennis traced them blind to the status of anybody. And he ended up doing it twice, actually, because we were surprised by the findings. And what we found was that there's the hippocampus there, you can see it, the frontal lobes. Hippocampus is deep, in the deep part of the brain. In chronic schizophrenia, we saw the reduction. So the straight line, which is 0, is the normal, and it's what we're looking at is the deviation from normal. So those with chronic schizophrenia showed left and right hippocampal reduction. Those at first episode, only the left, and those who were in the first 6 months of illness, it was trending down on the left, but not significant. And more importantly, the high risk didn't show that difference. So it didn't seem to be a marker. And we thought, well, that's a bit odd, that's funny. And this led to looking at the brain longitudinally, dynamically, what's happening over time, because there was a suggestion of a longitudinal trend, but of course we needed longitudinal data to show that. So this was the first study published in which we demonstrated that the frontal and temporal lobe areas, but also the cingulate, were showing a progressive loss of grey matter at the point of illness onset, at the point that psychosis began. And that led us to start really trying to map things longitudinally, dynamically, what is happening dynamically. And I think it speaks also to the clinical way that we look at our patients. We don't make a diagnosis on a one-off assessment, usually, especially early on in the illness. We need the passage of time to understand the progression, the evolution of the illness. And I think when we're looking at things like brain scans, it's no different. So here was a study over 2 to 4 years, baseline scan early in the onset of schizophrenia, then again 2 to 4 years later, and a comparison with normal healthy controls. We see in the healthy controls frontal shrinkage. And then in those with first episode schizophrenia, that shrinkage is occurring similarly, but about 2-fold increase. So it's going twice as fast as it should be. We think this relates to synaptic pruning being accelerated much faster than it should be. Indeed, when we looked across the whole brain, it wasn't just in the frontal lobes, it was actually a whole brain affair. So that increase, that 2-fold increase, was a whole brain reduction, suggesting and the possibility that actually the disorder is a whole brain disorder. It's just that the frontal lobes are maturing in adolescence, early adulthood, maximally, and that's where you see the significant changes. So we started thinking about developmental trajectories. We certainly see that evidence of progression. We see evidence of neurodevelopmental arrest. And we also have some evidence for neurodevelopmental lag. And what I mean by that is where there's an abnormality which then corrects itself and things get back on track. So we did this study together with Nitin Gogte and Judy Rappaport from NIMH in Washington. They had collected data over about 30 years on children with schizophrenia. They'd flown them in from across the country in the US. And together with Andrew Zaleski, an engineer that we employed, we looked at brain connectivity. So in the y-axis there, you see a measure of connectivity. X-axis, again, we see age. And these are longitudinal scans. So each dot is a second, third, fourth, fifth scan. Now, the blue line at the top are the normal, healthy children, typically developing children, showing their connectivity, a good measure of connectivity. The red line are those with childhood schizophrenia. So these are kids at around about age 12, around the time of puberty, that have developed psychotic symptoms. Really quite severe. Many of them end up on clozapine, and I've seen videos of these kids. They look very much like the patients on my unit. What's interesting, though, about this graph is the black line. So the black line are the siblings of those with childhood schizophrenia. And when you look at age about 12, the siblings and those who have the disorder look very similar in terms of this connectivity measure. But as you move into mid-adolescence, typically in the siblings, they catch up to the normals. So that abnormality seems to be corrected. So it raises questions. Well, is there some kind of resilience factors acting? Why do they manifest this abnormality, but then it seems to improve and get better? And this is another way to demonstrate this change. So on the left are those with childhood schizophrenia, and on the right the siblings. And the numbers of the various ages when the scans were taken. And you can see that there are more abnormalities in the childhood schizophrenia. And on the right, you see abnormalities early, but they normalize. They go back to normal. And I think this is a really interesting issue. And I show you this study because I think it's important in the context of the earlier discussion, the importance of the environment, the importance of socioeconomic level, and the importance of parenting. And I know that parenting is important to many of us here in this room. So Sarah Whittle was doing her PhD on this study and has now been running this study. And what the group did was to look at, and what we did was to look at social disadvantage and look at brain development across adolescence in terms of social disadvantage. And social disadvantaged also disadvantaged the development of the brain. So the maturing brain was impacted by a lower socioeconomic status. There is some good news, though, because in this study, they also looked at the influence of positive parenting. And positive maternal behaviors overcame this adversity in terms of brain development. The way they looked at parenting was to get the child and the parent to undertake a problem-solving task. This was examined and rated blind by the group in Oregon. And they demonstrated that positive maternal behaviors were able to ameliorate the adverse impact on the brain of social disadvantage. This is quite important because it means that we can make a difference. Parents can make a difference. Educators can make a difference in terms of what is happening in terms of brain maturation. And it led us about a decade ago to be working with Emeritus Professor Bruce Tong, an eminent researcher in autism, and Renee Tester, a neuropsychologist working alongside, working with children with autism-related or spectrum disorders. And Bruce and Renee had noted that some of the children that were being diagnosed with autism didn't really fit the category properly. And in fact, they seemed to have features of schizotypy that we heard about from Vaughan earlier today. Importantly, they tended to develop preoccupation with internal fantasies. They became preoccupied with these fantasies to the point where they couldn't concentrate in school, they couldn't undertake the tasks that they wanted to normally, they wouldn't engage socially, etc. So they looked autistic, but when we looked at them in detail, whilst they shared some features with autism, there were important differences. And indeed, we've developed the Melbourne assessment of schizotypy to be used in children around 8 to 12, so that we can actually distinguish them from kids with autism. And we've now been looking at brain structure, brain function, and also cognition. And all of those differ in the kids with schizotypy. And this is a typical example. Often Renee and Bruce will get them to draw a picture about things that they're thinking about. Ruth here depicts a picture of distressing perceptions. She has the voices of monsters in her head, and she's asked the clinician to help by writing on the picture, push them back into thoughts and memories and make them not real. So really very distressing for this young person. So we're now looking at those kids in a lot more detail in terms of their brain function, and Cassie Wannen is working with me on that, on those studies. So the brain is changing, it's dynamic. We shouldn't be thinking about it in a static way, but also we need to place it within context, within a brain maturational context, if we're going to understand what's going on. Resilience factors are likely to be important. And then there are a number of other factors that we need to be looking at. How do we explain the brain changes? What is it that is causing these brain changes? Be it synapses, be it axons, be it grey matter. So I'm just going to focus now on a few of the studies that we've been undertaking, and focus particularly on our recent findings with metals, because I think they're particularly exciting. First of all, I will show you the latest genome-wide association study of schizophrenia. This is the largest genetic study that, again, we contributed data to from across Australia. And so there are 76,700 plus cases and 243,000 plus controls in this study. Huge study. And you need these large numbers in order to identify each of those peaks, because the effect of each of those peaks is very small. And to achieve significance for a genetic study, you need huge numbers. What we see there is called a Manhattan plot, because it looks like the Manhattan outline. And each peak represents, that is above that red line that you see there, represents a genetic locus that is genome-wide significant. In other words, it's contributing to the genetic makeup of schizophrenia. And what you can see is that it's across the whole genome. It's not just one gene, though there is that large peak. And that large peak is at a very high significance level, P of 10 to the minus 39, if you can believe it. So highly significant. And that is pointing to immune function and pointing to inflammation. So the data that we've heard today about inflammation is certainly important and relevant. And the immune function is relevant also to the activity of the brain's immune cells, and also to the system that modulates those and modulates synapses in the brain, which, as I said earlier, is what we think are affected in schizophrenia. This study goes a step further. It used some impressive techniques to try and explore the meaning behind some of these genetic markers. And I just want you to take note here, because what it points to is neurons. It's saying that the genes that we've identified here are picking up abnormalities genetically that are relevant to neurons, not to the glia or to the other cells that are in the brain, but rather to the nerve cells themselves. And not only that, that the site of pathology is not restricted to a particular area of the brain. In other words, it's more likely to be global, consistent with some of the data I showed you earlier from our brain imaging. So it suggests that there's a genetic underpinning to the brain changes we might be seeing, that neurons are implicated. And not only that, drilling down further, it's particularly the synapses that are relevant, speaking back to the notion that synaptic pruning is an important factor in terms of schizophrenia pathology. We undertook a study that took about 5 years to do, and Maria Dibias did a fantastic job here. We were interested in the brain's immune cells, the microglia they're called. And after that 5 years, we had a negative study. What we found was that the microglia didn't show evidence of activation. We thought that if inflammation is important, there'd be activation of the brain's immune cells. We didn't see that. If anything, the levels were slightly lower. Now, this is a negative study, but a negative study is so important. So often we have positive study after positive study in schizophrenia. That doesn't help to guide us. Negative studies are really important and must be published because they point us in the right direction, away from things that we believe to be happening but are not actually happening. So it led us to be looking at some other systems in the brain, and I'm just going to touch on two of those. The ubiquitin system in the brain, we had some evidence from our own genetic study that the ubiquitin pathway was implicated in schizophrenia. Now, the ubiquitin pathway is important in terms of eliminating debris from cells. So it's like the trash can of the brain. If you accumulate debris in your cells, they'll become clogged. You need to get rid of that debris if you're going to have the cell functioning properly. The ubiquitin system, and it is a whole machinery, is important in removing protein that needs to be removed. What it does is to tag the protein and then sequester that and then remove it from the cell. So we looked at the machinery, and we looked at it in both blood and brain. So this is a post-mortem study as well as a blood-based study, and we found that the tagged proteins, those tagged with ubiquitin, so the ubiquitinated proteins were elevated in the blood and brain of patients with schizophrenia. We looked at the machinery and how the machinery was working. That seemed to be working okay, or at least the bits that we looked at. So it suggested to us that something else is going on, and that perhaps something called oxidative stress may be important in the disorder. And I know Michael Burke has been very interested in this and been undertaking studies to try and rectify oxidative stress. So what's oxidative stress? Well, the brain is basically running a whole bunch of chemical reactions. And it's important that they're in balance. If they're not in balance, then you have a system that could do damage to cells. You have a number of reactive species that are constantly being created, and you need to get a balance between oxidation and reduction in terms of that. And if you have oxidative stress in terms of these chemical reactions, you get damage to cells. So we were interested, therefore, to look at some of the factors that are relevant to oxidative stress. And iron is an important part of that equation. So why look at iron in the brain? Well, iron's been implicated in dementia, such as Alzheimer's dementia. Ashley Bush at the Florey Institute has undertaken a lot of work on this together with Sandra, and we work together to look at the patients, to look at post-mortem brains of patients with schizophrenia. So it's the most abundant metal in the brain. It's essential for proper functioning of cells, neurological processes. It's involved in oxidative metabolism and therefore likely to be relevant here. It's also important in terms of dopamine. So it's important in terms of the receptors that have been implicated in schizophrenia. Dopamine is the key transmitter that's been implicated in the disorder. And it's involved in the formation of these free radicals that can lead to oxidative stress and neuroinflammation and mitochondria dysfunction as well. The other thing about iron is you need to regulate its level, especially in the brain. So the brain has a number of mechanisms to try and keep the balance just right. Otherwise, you're going to be in trouble. And the way it does that is there's another protein called ferritin, which acts like a cage to take the iron and keep it safe so that it doesn't do damage to the brain. So if you disrupt iron levels and ferritin levels, you're likely to get some kind of damage happening. The blood-brain barrier, so the barrier between the brain and the rest of the body is an important factor here as well, and is important in regulating what is happening between brain and the rest of the body. So we undertook this study. You can see there the numbers of patients and controls. We used data from 3 different brain banks, including here in Sydney and working with Cindy Shannon Wyckett, and also from the US, as well as the Victorian Brain Bank. And we were able to look not only at iron, but we looked at all the metals, but iron came up as the most relevant. So if you look at these graphs here, the left one shows elevated iron levels in the brains of patients with schizophrenia. The middle graph shows reduced ferritin levels in the brains. In other words, that which protects the brain from the iron is reduced and the iron is increased. And the third graph is the ratio between the iron and the ferritin. So potentially we have a toxic environment for the brain for patients with schizophrenia. The other thing was that when we were writing this paper up, and it was published recently in Molecular Psychiatry, a Nature journal, I said to them, to Amit Lotan, who's the first author, I said, he was writing it very much in the mold of a neurodegeneration paper. I said, Amit, this is not a neurodegenerative disorder. Schizophrenia is not like Alzheimer's dementia. I said, look at the data by age. Let's see what we get. And when we do that, what we find is that the elevated iron levels are particularly so in those below the age of 35. If you go back to our brain imaging, that's the kind of, it's that early age when the brain changes are occurring maximally. That's when the grey matter loss is occurring. And similarly, the ferritin is lower in that early phase. So this early period after the onset of the disorder is important. And if we're going to make a difference, we really need to be targeting that early phase of the disorder. We didn't stop there. We're now looking at brain imaging to try and index iron in the brain. And Pasa and Ravanfa, who is undertaking his PhD and now training in the States in psychiatry, and Werda Saida, who's a very talented engineer working on brain imaging. We were able to use an imaging technique to index iron in the brain. What you see there is elevated iron levels in a part of the brain that is rich in iron, so it kind of validates the notion. But we did see elevations in iron in substructures of the brain. And then again, we saw a difference, when we looked at the relationship to brain volume, it was in the frontal lobe and the hippocampus where there were differences between patients and controls. So we think that's very exciting data because it may point us in ways to try and treat the disorder. It points us to be really thinking about that early phase of the illness, that this is a particularly important time. But all of this work, and I haven't been able to show you all of it, and I haven't focused on any of the cognitive work, but we've now moved to a preclinical model I'm not going to talk about this because Jess Nithyanantharajah is going to be talking about that next, but I'm just going to introduce it. So we were interested in a brain maturational model in the mouse, and we tried to match the developmental periods for mouse and human. I'm sure Jess will go over this in more detail, and I've only got about a minute left, so I better move on. But we're also doing brain imaging in those mice. And we're also working with this guy here, the tall guy, Arthur Christopoulos, who's the Dean of Pharmacology at Monash, who's an absolutely super talented guy. And we're looking at novel treatments to correct some of the abnormalities in our mouse model. This is one study we did where we were able to use antipsychotic drug together with a novel compound that acts on the muscarinic system to shift and correct cognitive deficits, but I won't go into the details. So I better finish. The brain is changing, it's dynamic. Genetics are important because they point to pathological mechanisms that may be relevant. And understanding things at the cellular level means we might be able to start to get a handle on new ways to treat the disorder. And that's clearly what we need. Looking at things over time is important. So longitudinal studies are key. Working across disciplines, absolutely essential. We need physicists, engineers, mathematicians. We're even working with a philosopher more recently. And that's just a fabulous way to go because it really expands the way that we think about such a complex disorder. And because it affects the very essence of who we are, it affects our conscious being, we need all of those disciplines to play. Now, there's one further thing I want to say in the last minute. Certainly, we've had a lot of collaborators. We've collaborated across Victoria, across Australia, and across the world. And it's been fabulous to visit all of these places, to meet with, very talented people and to work with them to really try and understand a disorder that has proved so difficult to get a handle on. And a number of consortia across the globe. Just finally, my own personal story, and this whole journey has been a personal story. And I see Jackie in the audience there, and you've really prompted me here, Jackie. So I want to make a dedication to my auntie, Frida Paraskevi Pantelis, who suffered with schizophrenia. That's my mother on the left and Paraskevi on the right. This was Paraskevi when I was at uni. I had a lot more hair then, of course. I was a bit thinner too, actually. This was her a little bit later on. She died in 2014. But interestingly, she lived in Moonee Ponds. She was hallucinating constantly. She had ideas that the Russians were implicated in some conspiracy. I had her seen by a number of psychiatrists. We couldn't put her on an order because it wasn't appropriate. So she was unmedicated for 50 years. And she functioned very well. You can see she's got a really good affect, actually. And she was very personable, but had all these experiences that were going on. My parents looked after her. They wanted to normalize her life. They struggled to normalize her life. And of course, that was very difficult. And then my family, including my children, have continued to look after her for many years. And these are my parents, and we've created a scholarship in their name. And these are some of the recipients, including Cassie, who'll be speaking soon. Thank you.